Question number 18. The morphological novelty of echinoderms, marine invertebrates with radial symmetry, usually starlike, around a central point, impedes comparisons with most other animals, in which bilateral symmetry on an anterior-posterior head-to-tail axis through a trunk is typical. Particularly puzzling are sea stars, thought to have evolved a headless layout from a known bilateral origin. Applying genomic knowledge of Saccoglossus kowalevskii acorn worms, close relatives of sea stars, and thus expected to have similar markers for corresponding anatomical regions, to the body patterning genes of Patiria miniata sea stars, Laurent Formery et al. Observed activity only in anterior genes across P. miniata's entire body and some posterior genes limited to the edges, suggesting that blank. Which choice most logically completes the text? A. Despite the greater prevalence of anterior genes in sea stars' genetic makeup, posterior genes active at the body's perimeter are primarily responsible for the star-like layout that distinguishes sea stars' radiosymmetry from that of other echinoderms. B. Contrary to the belief that they evolved from early ancestors with the bilateral form typical of many other animals, sea stars instead originated with an atypical body layout that was neither bilaterally nor radially symmetrical. C. Although the two species are closely related, there is only minimal correspondence in the genetic markers for head, tail, and trunk region development in P. miniata sea stars and S. kowalevskii acorn worms. D. Rather than undergoing changes resulting in the eventual elimination of a head region in their radial body plan, as previously assumed, sea stars' morphology evolved to completely lack a trunk and consist primarily of a head region.
So in 18, we are talking about the morphological novelty of echinoderms, that is the marine invertebrates with radial symmetry, usually star-like around a central point. And how to compare them, because this impedes comparisons with most other animals in which bilateral symmetry on an anterior-posterior head-to-tail axis through a trunk is typical. So if you take a human body also, here from top to down, this is the left and right. Let's say this is the left and this is the right. So this is the bilateral symmetry which we have. Whereas these starfish, that's why they are puzzling. Particularly puzzling are sea stars thought to have evolved a headless layout from a known bilateral origin. So that's what they think, it's a headless layout because we cannot see the head. Applying genomic knowledge of Saccoglossus kowalevskii acorn worms. These are the close relatives of the sea stars. So they have now applied genomic knowledge to study this, and thus expected to have similar markings for corresponding anatomical regions to the body patterning genes of Patiria miniata sea stars. So Laurent Formery et al. Observed activity only in anterior genes across P. miniata's entire body and some posterior genes limited to the edges, suggesting that blank. So which should we choose? So we have to go with answer choice D. This is a little difficult to understand, but let's see. So choice D is the best answer because it most logically completes the text discussion of the morphology, form and structure of sea stars, a type of echinoderm. The text indicates that echinoderms have radical symmetrical body plans symmetrical around a central point, usually in the form of a star, whereas most animals have bilaterally symmetrical body plans symmetrical along an axis running from head to tail through a trunk. According to the text, sea stars are unusual echinoderms because despite their radial body plan, they descended from known bilateral ancestors. So they have descended from bilateral ancestors. This shift of body plan was thought to be a process of losing the genetic markers associated with the head region. So if you look at the starfish, you might think that it doesn't even have a head. But what happened when they checked the genes? The text explains that by comparing the genes of one sea star species, P. miniata, to those of a close relative, the acorn worm, researchers determined that instead, anterior the head genes are active across the sea star's entire body. So the head genes are active across the entire body, whereas the posterior tail genes are active in limited peripheral locations of the body, and no trunk related genes are active. So what does this suggest? This suggests that rather than becoming headless as they evolved from bilateral ancestors, sea stars developed a body plan consisting almost entirely of a head region, with a minimum tail region and no trunk region present. So they have entirely or almost very limited posterior genes. Only, their entire body is having the anterior genes, which is related to the head. And no trunk and tail region genes are found at all. So that's why we have to go with answer choice D, based on the genetic research which is done. And choice A is incorrect because the text doesn't identify how any particular region of sea star's bodies influences the layout of sea star's radial symmetry. Moreover, the text indicates that the radial symmetry of echinoderms is usually star-like, not that a star-like layout distinguishes sea stars from other echinoderms. Choice B is incorrect because the text doesn't suggest that the idea that sea stars evolved from an ancestor with bilateral symmetry is incorrect, describing the bilateral origin as known and doesn't address any body plans other than those with radial or bilateral symmetry. The text strongly suggests that rather than revealing something about sea stars' origin, Formery et al.'s findings contradict the assumption that the current body plan of sea stars is headless. So that is getting contradicted. First, they think it is headless, but actually, as per the genes, it is having only the anterior genes. Choice C is incorrect because the text suggests that Formery et al. Were able to make determinations of P. miniata sea star's body plan based on the comparability of genetic markers between P. miniata and S. kowalevskii acorn worms. The text indicates only that little or no activity was observed in certain types of genes associated with body development in P. miniata, not that those genes turned out to largely differ from body development genes in S. kowalevskii. So I hope that's clear.